today we will finish up the chapter 9, control and regulation, which will be the plant hormone topic. So last week I already covered the nervous system and also the animal hormone, the human hormone too, the mechanism about the steroid and non-steroid hormone. So today we will focus about the plant hormone. Okay. So in our syllabus, the plant hormone that we need to learn will be auxin, gibberellin, cytokinin, and also ethylene, and also abscisic acid. So there are 5 hormones you need to learn. So in SPM you learn only 2 hormones, right? I think in SPM you only learn about auxin and also ethylene only. But in STPM you need to learn about the 5 hormone. Okay, so the first hormone that you need to learn is auxin. So I think the auxin hormone is very famous for you guys, and also familiar to you guys lah, because you already learn in SPM too. So as you guys know, auxin is play the role in the growth. You know, like the auxin is the play major role in the growth, and there are 2 types of the mechanism that they can respond toward the light. Either it is respond toward the light or respond toward the gravity. So when the growth of the plant that respond toward the light, is phototropism, right? And then when the growth is growth toward the gravity, then we will call as geotropism. So normally auxin is produced in the apical meristematic plant. So if you still remember that in biology semester 1 that you learn about the meristematic tissue, we know that meristematic tissue is located at the bud, at the bud of the shoot or root, right? Okay. So normally auxin will be secreted at there. So there are no specific location. The specific location is only at apical meristematic cell only. And then normally it is transported from cell to cell, toward all the plant that require the growth of the plant. Okay, so normally this is what I already mentioned about when the growth of the plant that using this hormone toward the light, we call as phototropism. And then we know that in auxin in shoot, the cells on the darker side will elongate faster than brighter side. Okay, so you will see this diagram. So normally this is phototropism, because this is the plant growth of the plant toward the sunlight. So this is the shoot, apical meristematic of shoot. So we know that auxin is accumulated at here. So normally in sunny day, the auxin molecule will be accumulate at the darker side one. It would accumulate at darker side and elongate at the darker side part. So that is why that you can see that the plants are faced toward the sunlight. You see this. Why? Because the auxin is accumulated at the darker side and they will only elongate at there. The cells only elongate at the darker side. Darker side, the cells does not elongate. It only maintain at there. But only this one, the cells are elongating. So that is why that you can see that why the plants will face toward the sunlight. So this is the reason lah, auxin. And another one is about the geotropism. So there are 2 types of geotropism, but positive and negative. So normally if you still remember that geotropism is the response of the auxin toward the growth of the plant based on the gravity. So you see here the auxin is accumulated either in shoot and root, right? So in root, especially in root, the auxin will face toward the gravity. It will accumulate based on the gravity. So in root, it will accumulate at the lower part only, similar as the shoot. It will accumulate in the under layer of the shoot only. But the difference is auxin stimulate growth in the shoot. So when the auxin is accumulated at the under layer of the shoot, it will stimulate the growth. But in root, the auxin function opposite way. It will inhibit the growth. So meaning that only the upper part of the root will be growth only. The upper part will be elongated only. So it is totally two different function. Auxin in shoot is stimulate the growth, but auxin in root will inhibit the growth. So when you talk about the function of auxin, we normally talk about the shoot only, because root will inhibit the growth one. Okay, so this is very important that you need to know. Okay, and then you still remember that I just recap from the SPM. I think SPM you already learned about the experiments that prove about the relating about the auxin molecule. So normally one of the experiment is conducted by the Charles Darwin. So normally this experiment is to determine phototropism of the auxin. Whether why we say that the auxin will be faced toward the sunlight one. Why? So normally these scientists use the 5 parameters to conduct the experiment. So the first experiment is the control meaning that all the plant shoots are not be cut down, or we call as collotype. This one is collotype. So meaning that the collotype is maintaining, we act as control we not change any part of the plant structure at there. But in second diagram, we remove the collotype. Okay. Once we remove the collotype, they found that actually no any response at all, meaning the shoot also cannot grow. No shoot will be growth. The shoot is maintaining at here only. Not meaning that the plant is die, but the plant cannot grow. It only can maintain the height of the shoot like this only. So this meaning that auxin is highly concentrated in the tips of the collotype, or in the tips of the shoot. So if you cut down the tips of the shoot, then no auxin produced, so that the plant cannot grow anymore. And then you see this diagram. When you cover the collotype with the cap, or we known as the opercle cap, meaning that this is totally black cap. No untransparent cap. It is totally dark cap that can totally inhibit the light enter into the plant. So that since there are no plant, there are no plant, no sunlight at there, so there will also no any response to, because auxin only respond when they are sensitive to the light only. So if no light respond, the shoot also cannot grow. But when you cover with the transparent cap, meaning that the light still can pass through the cap into the collotype, so that the auxin can be will have a response. So the auxin will like the positive tropism. It will accumulate at the darker side, and then promoting the cell elongation at the darker side. And then the fifth diagram is they cover at the base of the stem of the plant, rather than on tips of the plant. So when they cover in the on the stem of the plant, they found that the response still occur, similar to the transparent cap, totally similar. So meaning that there are still the plant light response toward the by the auxin, which the plant face toward the light. So they are still respond by the auxin too. So in this experiment, they conclude that auxin can be found in the tips of the shoot only. It only can be found on the tips of the shoot only. Okay, and then when and the first conclusion, and then the second conclusion is, when although you remove the all the tips of the shoot, you still tip of the shoot, but no light toward the auxin, the auxin still cannot have a light response for phototropism. So this is the conclusion that concluded by the Charles Darwin about the phototropism of light response from auxin. Another experiment is by Jensen. Okay, so normally this one is they using the any barrier that can be cut, that can be inserted unilaterally, or using the gelatin barrier inserted subapically. So you will see this diagram, meaning that they inserted this mica barrier is directly inserted into the tips of the plant, but only half only, not totally cover the plant, but only half. So you see here when this arrow is indicating the light response. So you see here when there are the barrier at here, meaning that when the light is come from the right hand side, there are no response at here. And then if you see this, when the barrier is inserted at the right hand side and then the light is come from right hand side, but they are response at here. The results show that they are response at here. So meaning that auxin only can accumulate at the darker side and provide elongation toward the light response. But for this one, since it is already barrier at here, they already got barrier at the darker side, so that auxin cannot accumulate at here, so no light response by auxin. And then this one is they use the gelatin barrier. So normally this one is they totally cut, and then like previously experiment, if you totally cut, they will totally cut and then they cover by the gelatin barrier. So this one is inserted, they just cut the portion of the tips of the plant and then they insert half only. But this one, they totally cut down the tips and then they insert the gelatin barrier. Okay, after that, they found that they are response. They will have a response toward the light. Meaning that if you using the gelatin molecule, meaning that the auxin still can accumulate at the darker side and then still stimulate the growth of the plant, compared to using mica barrier. If you're totally using mica barrier, meaning that mica is the totally blocked, right? You totally block it and then no response at all. Okay. Another experiment is by Vance experiment. Okay, so this experiment is where they're using totally cut down the tips of the plant and then they transfer the auxin to the agar, to the jelly. And then later that they'll transfer this jelly already high in concentration of auxin, transfer back to the plant again. So they transfer back to the plant again without the original tips, but using the agar block that containing the auxin serum. So after they transfer back again, they found that Auxin block cause bending of shoot not exposed to light. So meaning that the hormone from tip is stored in agar block, and then the agar block put on colluted on the side, and then the colluted will bend toward the light, and then auxin move down the shaded region for response to growth. So they found that either you using the right hand side or left hand side, you cover the right hand side and left hand side, but if they are the light, they still can respond to the light. Cells on the shaded side elongate in response to the hormone. Okay. And another one is experiment by Bridge. Okay, so normally this one is they are using the similar, they just modify the experiment by Vance, but the difference is the auxin tips that the shoot tips that you use is they were extracted with the light first, and then they use the barrier to block the auxin development. Okay, so meaning that you will see here, you will see this diagram. So when the barrier is auxin block, so when they are the light from the left hand side, so if you still remember that auxin will be accumulate at the darker side one. So meaning that they will only accumulate at the big B part B only. The auxin will be accumulate at the part B region only. So meaning that part A region is less concentration of auxin serum. So when you use this block, agar block, transfer this agar block toward the plant again, you will find that the curvature of the plant for A and B are different. B will curvature more curve, and then A the curvature is very low. The degree of angle of curvature is low, but for this one the degree of angle is high. So why? Because B containing high concentration of auxin, so it will more highly respond toward the light compared to A. Okay, so this is the bridge experiment that want to tell you that the light response of the auxin. So even though in the agar block, not whole plant, but the auxin will respond only in the darker side, not at the light side. Okay, so this is all the experiment that related to auxin lah. So although syllabus does not mention, but you should know too, because in SPA you already learn. So whatever maybe in Form 6, and then they come to tell you, okay.